Today we will be presenting MediShare, a healthcare initiative focused on improving access to reliable diagnostic testing in developing regions. Our project aligns with two Sustainable Development Goals, SDG 3, Good Health and Well-being, and SDG 10, Reduced Inequalities.
Infectious diseases and antibiotic resistance disproportionately affect low-resource regions, yet these are the places with the least access to diagnostic testing. Most testing is centralized in high-income countries and relies on cold chain transport, making it slow, expensive, and often inaccessible. As a result, many communities lack local testing capacity, as well as scientific training and medical autonomy. This leads to delayed treatment, limited disease monitoring, and a continuing cycle of global health inequality. Our project would center around creating innovative packaging for our ELISA diagnostic kit, which would be heat-resistant and sterility verified. To ensure that these kits are sterile, we would include a built-in sterility indicator. This would be a color-changing chemical strip similar to those used in autoclaves, which changes color if the kit has been exposed to heat or contamination. It provides an immediate visual confirmation that the components are still safe to use, enabling quality control even without a traditional laboratory. Another key aspect of our project is the training. Our implementation would follow the train-the-trainer model, which is already used in global vaccination and malaria programs. Healthcare workers begin by working with a small group of community health workers, who then pass that knowledge on locally so that the skills remain within the community. The training goes beyond simply running the ELISA test. It focuses on understanding the results, recognizing when a patient needs to be referred for further care, and also building awareness of antibiotic resistance so that the test leads to informed and responsible treatment decisions.
Most ELISA kits must be stored at temperatures between 2 and 8 degrees Celsius, requiring continuous refrigeration. This dependence on cold chain transport presents significant barriers in regions across Africa, Asia, and rural communities where electricity supply is inconsistent and ambient temperatures frequently range from 30 to 45 degrees Celsius. The primary biological components of ELISA tests are antibodies and enzymes, both of which are proteins with highly specialized three-dimensional structures. These structures are maintained by weak intermolecular forces, including hydrogen bonds and ionic interactions. When exposed to elevated temperatures, these weak bonds break, resulting in protein denaturation. As a consequence, antibodies lose their ability to bind to target antigens, while enzymes lose their catalytic function and can no longer generate the color-changing signal required for detection. This structural breakdown renders the diagnostic test inaccurate or ineffective. To address this issue, MediShare integrates a 4-part heat stabilization strategy. This includes lyophilized antibodies, trehalose stabilization, vacuum-sealed barrier packaging, and lastly, thermal insulated outer packaging. Trehalose, a stabilizing disaccharide, is added to the antibody solution prior to drying. Trehalose replaces water molecules surrounding the protein and forms hydrogen bonds with the protein structure. During drying, it creates a protective glass-like matrix that prevents structural collapse and preserves the protein's three-dimensional integrity. Following stabilization, the solution undergoes lyophilization, or in other words, freeze-drying. The process involves freezing the solution and removing water through sublimation under reduced pressure. This results in a dry, stable powder form. By removing water, we significantly reduce the rate of heat-induced degradation and limit chemical reactions that would otherwise destabilize the proteins. After lyophilization, the sealed vial is placed inside a moisture barrier pouch. A desiccant is included to absorb residual humidity. The pouch is then vacuum sealed to remove air and heat, sealed to prevent environmental exchange. This stage protects the product from moisture, oxygen exposure, and contamination, all of which accelerate protein degradation at elevated temperatures. The vacuum-sealed pouch is then placed inside a thermally insulated container. The container is lined with low thermal conductivity foam to reduce heat conduction. A reflective inner layer minimizes heat transfer through radiation. The product is then positioned centrally to avoid direct heat contact, while the outer container is sealed to reduce air exchange. This layered insulation system slows external heat penetration and buffers against temperature fluctuations during storage and transportation.
MediShare supports SDGs 3 and 10, good health and well-being, as well as reduced inequalities, making diagnostic testing more reliable by stabilizing biological components against heat, ensuring sterility, and designing a more intuitive kit, thus reducing the risk of inaccurate results or contamination. This allows healthcare providers to perform testing confidently, even in low-resource settings, which strengthens local healthcare systems.